Hello everyone, this is Jennifer Nesta and we are back for Bioremediation Part 2, which is MIC 3732, Unit 5, Environmental and Agricultural Microbiology. So we have already covered what is biodegradation, and we are here to learn the rest of the bioremediation methods. Okay, shall we begin? So we have already seen in part one that we have lots of different types of remediation, environmental remediation techniques, and they are divided into two parts, which is ex-situ and in-situ. And ex-situ practically means taking the sample away to an external place. So you have a polluted site, you have an kilogram, you have different tons of soils, and you take the soil away to a treatment plant, or you pump the water away from to a particular treatment plant, you treat it there and you release it back. So this is ex-situ techniques, and for in-situ, these particular remediation techniques are applied in place. Wherever the polluted site is, we apply the techniques there. So ex-situ primarily involves excavation, that is, you dig up the soil, you treat it, and you bring it back. And similarly for groundwater, it is applicable for soil, and for groundwater you pump them and treat them. So for in cases of underground water aquifers and underground subsurface soil sediments, you can use multi-phase extraction techniques wherein you extract contaminating gas, pollutants, or metal elements. So again, in case of contaminated aquifers, you can enhance the production of surfactants, or provide surfactants, make amendments in a treatment plant, and remediate it. So, and you can also add oxidating agents, and or treat it with electrokinetic radiations, or use stabilizing agents, chemical agents to stabilize the soil so that the soil does not denature any further. So you have these various physical chemical techniques, and bioremediation is just one of them. So you have to keep in mind that physical chemical techniques are one of the most used techniques for environmental remediation. So we are not at a phase where bioremediation is widely used. So in almost all the industries, whether it is effluent industry, paint industry, pesticide industry, these industries, the treatment plant, the effluent treatment plant usually uses some kind of physical chemical technique rather than a bioremediation technique. So you can say that bioremediation strategies are applied in several places, obviously, but most often bioremediation strategies are still under development. It is a heavily researched place, and R&D emphasis on this particular field is very high. So under bioremediation techniques, we have bioaugmentation. Bioaugmentation and bioremediation is practically similar. Bioaugmentation is the introduction of a genetically engineered bacterium. So in bioremediation, it can do two things. It can either stimulate uh, the indigenous population, which has the capacity to biodegrade an organic pollutant, or else you can engineer a vector or a bacterium which has enhanced, advanced capabilities of biodegradation or biosorption, and you introduce these lab-generated bacteria into the environment. Similarly, you can do the same for microremediation. You can use fungal consortiums, you can use plants, which is phytoremediation. So again, in the next slide, it is the same thing that has been given in a different format. You have ex-situ and in-situ techniques. So you can see that there are different particular names, which is biosparging, again bioaugmentation, biological reactive barriers, composting, construction of wetlands, slurry and aqueous reactors. You have biopiling. So these are all there are lots and lots of bioremediation techniques, and in the upcoming slides we are going to discuss just a few of them which are being used currently worldwide. So okay, so in ex-situ bioremediation, some of the techniques which are used very widely is land farming, bioreactor, composting, and biopiling. So, as it says, ex-situ techniques are applied to those samples of soil and water which are removed from the site. So they are removed from the place of pollution, they're excavated or pumped out, and they're taken to another place. So when a larger surface area of water or soil is polluted, it is not virtually possible for us to excavate or pump out the whole system and remediate it in a treatment plant. So obviously, ex-situ is suitable only for very small projects. So only certain tons of soil can be remediated, or certain amount tons of water gallons of water can be remediated. So it is applicable, and it is well good and done for smaller projects. And these techniques, as you see on the screen, it consists of land farming, using a bioreactor, and composting and biopiling. So in the upcoming slides, we'll see what these techniques actually are. Okay, so biopiling. So biopiling, you will also observe that in the upcoming slides that these techniques are more or less the same, just the types of setups are different, and the parameters that are involved are a little bit different. But most often, the phenomena we find is very, very simple. It is mostly based on aeration and providing nutrients. So, as you can see in the diagram, uh, you have contaminated soil. You have contaminated soil all over, and along with that, you have pipes perforated. It says it is a perforated pipework. They are merely pipelines which has holes in them, and through these holes you give air, that is, you give O2, oxygen, you give this O2 for metabolism, and you excavate soil from a contaminated area, you pile them over these pipelines, and you cover them with a propylene cover, polypropylene cover. And one more thing that you provide other than oxygen is nutrient supply tank. So as I said earlier in part one, biodegradation is a very, very slow process. It most often it results in complete mineralization of the organic pollutant into carbon dioxide and water. So to enhance but bioremediation, as we discussed, is an engineered process. We're going to enhance biodegradation, so we have to fasten up the process of biodegradation. And how do we do that? So in an ex-situ technique, we have taken tons and tons of soil, we have piled it over, so that is biopiling. We have piled it over a perforated pipe box, and this contaminated soil, say for example it is contaminated with oil and or hydrocarbon, so these soils already have an indigenous population of microbial communities which are capable of using hydrocarbons or oil as a substrate and degrade it and use it for energy, produce ATP, and give up carbon dioxide. They already have and indigenous microbial communities. They can also be called as autochthonous microbial communities. So all we need to do is provide them enough air. So in case of contaminated soils and waters, they are stagnant into place. Aeration is very difficult to reach. Not only that, these microorganisms inside the soils have to be very, very competitive to get a nutrient and mineral supply. So they already have a hard time dealing with contaminated pollutants, organic pollutants, which are very, very recalcitrant in nature. So here what we do is we enhance the procedure by giving them oxygen and also by giving them nutrient and water supply. So the process becomes very fast. So the particular pollutant, organic pollutant in this case, is degraded, is converted into water and carbon dioxide, and if at all it is not completely mineralized, there ought to be some kind of metabolite which could again be toxic or not toxic in nature, and whatever the metabolite that results in, it is collected at the leach gate collection slump. So if at all we have some contaminants, it is eluted by this water that you provide, and it comes running off to this particular leach gate slump and it's collected. So you can see that this whole pile, this piles and piles of soil is closed with an HDP line and a polypropylene cover because they want to stop runoff evaporation and volatilization. So once the soil is piled up, it could be trapped due to elements of nature like rain and wind. So you cannot afford the soil to transfer from one place to another. So obviously you add a carbon, and hydrocarbons, diesel byproducts, crude oil byproducts are volatile in nature. They evaporate into air. That can cause and along with biodegradation, that can cause toxic fumes to be generated, which will ultimately result in air pollution. So to control volatilization of oil and hydrocarbon contaminants, they use a polypropylene carbon. So this process of enhancing or stimulating the indigenous microbial communities is also called as biostimulation, that you see in the upcoming slide. So this is how biopiling is done in original setups. Okay. So we have the next technique, ex-situ techniques, which is composting. So most of you are familiar with how vermicomposting is done. We know that it is done using earthworms. We decompose organic matter into simpler substrates, which is readily available for plant growth. So here the concept is very same. It is very similar to biopiling also. It is adding aeration. It is increasing aeration by mixing soil and activating the indigenous microorganisms. Only the setup is different. So what is the setup and how is tons and tons of soil composted? So you see in the next slide. So if you see in the slide, we have soils piled up in rows. So this particular rows of soil is called as windrows. So they are piled into rows, and you have a special tractor like machine which is involved heavily involved in aerating. So as this tractor proceeds forward, what they do is they completely turn the soil over and over, and they also provide nutrient and water supplies. So when they keep on aerating the soil and providing water supplies, naturally the process of biodegradation again enhances by activating the indigenous microorganisms. So the phenomena behind both composting and biopiling is very same. The only way in which it is arranged is different. So here they have windrows, and they have perforated pipe works, and they pile it under a polypropylene cover. So that is the only difference between composting and biopiling. But the concept behind both of them are that they are ex-situ, the, and they use the indigenous or the autochthonous microorganisms. Okay, so when we discussed ex-situ techniques, we also saw that there are bioreactors, and we also saw that there are land farming. So we are not going to discuss them here because the land farming and the bioreactor techniques can be applied for both ex-situ and in-situ. So we will discuss them in a separate category while we move forward into in-situ bioremediation here. So again, in-situ is applied to soil or groundwater at the site. It is placed where the pollution occurs. So that is because we want to reduce the disturbance that the particular environment is subjected to. So imagine a place where wild animals live, where you have a sanctuary, but you have a pollutant contaminating the area. When you disturb a soil profile, so in ex situ, we are actually going to excavate or dredge up the soil, dig up the soil using JCBs, which are heavy machine operators. So when you disturb the soil profile is going to be disturbed, the horizons are going to be disturbed, and the soil horizons are produced by years and years of biological and physical and chemical weathering. So it takes lots and lots of years for the soil horizon to be developed. But at a fraction, within a fraction of a moment, we end up destroying the particular environment. So, to prevent disturbance to the particular environment, we try to apply in-situ techniques. But because it is all the resources for bioremediation has to be taken to the place of contamination. Naturally, we need specialized equipments because we are taking the remediation to the place, and because we need specialized equipment, this particular technique is going to be a bit more expensive than ex-situ, but it is, of course, it is minimally invasive to the soil. So what are the different techniques that we can see in in-situ bioremediation? You can see bioventing, you can see biostimulation, the bisorption, phytoremediation, and the biology. So we'll discuss these techniques one by one in the. Okay, so in situ bioremediation, the first concept that we are going to see is biostimulation. Stimulation. So this is practically what we have seen already. This can be applied for ex situ also, but when the particular soil or water resource cannot be transferred from the polluted site to a treatment plant, this is one of the basic things that we can do. So biostimulation is just addition of nutrients and providing air to the polluted site, so that, again, as we discussed, the indigenously growing microorganisms that are capable of degrading the pollutants will grow. So when the types of nutrients that you add can vary. So sometimes in case of metal oxidizing bacteria, we add different kinds of redox potential substrates, which will ultimately enhance the recovery of the metals from the polluted site. So here you can see how biostimulation works. So you provide nutrients. The nutrients are used by the indigenous microorganisms, which are capable of degrading your pollutant, and they produce carbon dioxide and water. So this is how biostimulation works in an in-situ condition. You have nutrient tanks. Here the nutrient they are giving is nitrates, so that they can enhance nitrate reducing and phosphate solubilizing microorganisms, and they provide H2O as an electron donor. So now this is the zone of contamination, and these nutrients are given to the groundwater table to induce or activate the indigenously occurring microorganisms so that they grow well, they metabolize well, and this zone of contamination is ultimately degraded down and it is leached down to the groundwater flow. So this is how this particular tanks are used, and aeration pumps, injection wells are used to deliver the nutrients and the electron donors to the contaminated site. So this is how biostimulation works. So the next concept is biosorption. Biosorption is what we have learned already in a micrometal interaction. It is primarily the absorption between a metal ion species against the biomass of a microorganism. So biosorption is known to occur, or it easily occurs in an aqueous solution. So this adsorption of metal ions on the cell membrane occurs at different it is pH dependent, and it is also dependent upon the equilibrium between the metal ions. So the degree of affinity is determined by the solid and the liquid phase. So how much of metal is available and how much of cellular membrane surface is available. These determine the amount of metal intake that is absorbed on the surface. So these are two different setups where you can manipulate biosorption into recovering metals from your effluent. So for example, let's say you have an effluent sample, a water sample which has metal ions still left in this, and you're going to add a biosolvent. It can be a microbial biomass, it can be living microbial biomass, it can be dead microbial biomass, it could be an immobilized sodium halogenate immobilized biomass. So it could be any kind of biosolvent that we use. Sometimes people use coir pit and lots of other biological materials which have shown considerable efficiency in sorbing onto metals. And this particular biosolvent in metal is kept in contact in agitation, and later on the biosolvent absorbs the metal and is filtered out. And this biomass can be used for recovery of the metals. So this is called as bath system. So this is a biosorption in bath system. And another kind of mechanism is continuous system. So in continuous system, as you can see here, you have polluted water, and this polluted water is pumped into a column. So this column contains various sets of algal bisolvent beads. These algal cells have been converted into beads, and they are packed on various layers. So this polluted water is pumped down. This algae absorbs the metal ions in the polluted water, and it is transmitted, and the treated water is taken out. So these algal cells can be later on transferred, and the metal ions can be recovered from the algal cells. So the concept is the very same. A biosolvent packed in a column, and you pump the wastewater through the column, and you collect it treated water again at the end. So this is how biosorption can be applied in two systems, which is batch and continuous. The next concept is bioventing. So bioventing and biosludging are very, very similar, and it is also very similar to biostimulation. So in biostimulation, we gave the contaminated site, we gave access to aeration, and we gave nutrition. So this is the same concept again, but the only addition is we give oxygen, we give air at a flow rate. So these are similar to agitators, where they keep on pumping air into the contaminated site along with the nutrients. So it is very, very similar to biostimulation. Only difference, or one of the major difference, is that it reaches much more deeper contaminated sites which are near to your groundwater table. So this is again a variation of biostimulation, and this also ensures that indigenous microorganisms are available to biodegrade organic constituents. The difference between biostimulation and bioventing will be its unsaturated source. So the depth is much more higher in bioventing than biostimulation. Okay. So yet another process that we use for in-situ bioremediation is bioleaching, which is also called as biomining. So most often an environmental site can be an used up mine. So these used up mines have been exploited so much that almost all the ores have been excavated already. What we have left is a low grade ore, but we can use biological interactions between microorganisms and the minerals. We can exploit and manipulate this particular microbial interactions to extract these metals from a low grade ore. Then now this is called as bioleaching or biomining, and it is very, very cheap in comparison to normal mining techniques that are used to excavate gold or copper or other minerals from mines. So what is the setup? So you have the body of the ore, we add in bacteria along with the liquid medium and the substrates. and this gets attached to the bacteria, attaches and binds itself to the metals, and it is pumped out. And what we call a leach liquor, and from this leach liquor, the metal, the valuable metal, the extracted from the low grade ore is recovered. Now that process is called biorecovery, which is again a very extensive uh, topic. So we recover the metal from a low grade ore. So that is bioleaching or biomining. Okay, this is yet another illustration of how biomining works. So we have a different physical and chemical process, and it reaches the downstream process where you add bacterial fungal inoculum, and it causes recovery of metals. Okay, so we are going to discuss what are the different techniques of environmental remediation that you can apply for both ex-situ and in-situ. So one of the topics that can be applied on the polluted site or on the excavated contaminated soil resource is land farming, and it is very similar to biofarming, only that the HDP layer is present, but what is not present is you don't have a polypropylene cover. So that is one major difference between land biofarming and biopiling. Okay, so the difference between biopiling and land farming is there are two different types of soils that is present here. So you can either add uncontaminated soil to the contaminated site, or you can take the contaminated soil, take it to a place where the contamination is not present, it is a clean site. You can add and mix the contaminated soil with the uncontaminated site, and uh, add a bioaugmented bacteria or stimulate indigenous microorganisms. So it is primarily uh, decreasing the density of pollutants in the soil by either adding new soil or adding the contaminated soil to uncontaminated soil. So this can be either ex situ or this can be either in situ. And again, the concept is the same as biodegradation. So um, we are just decreasing the concentration of pollutants and increasing the aeration for the biofarming. Okay. And this is illustration, well explained illustration from US EPA, published in 1994. So once a contaminated soil is added, soil is aerated often by tilling, and the groundwater metals, the particular contaminant is leached out to the groundwater levels, so they are always monitored, and the leachate, what we call the residual, is collected and again treated. Very basic setup for usually observed in biofarming. So bioaugmentation, we have already discussed bioaugmentation. It is the addition of non-native microorganisms. So biostimulation is inducing indigenous microorganism to work faster by providing nutrients and water and oxygen. Bioaugmentation is a completely different. We are going to genetically engineer a microorganism with higher efficiency and faster degradability of your pollutant. And we take this lab generated microorganism to the particular site of pollutant, or we take it to the treatment plant, we add as bioaugmented bacteria or fungi and make sure the pollutant is completely mineralized in a particular time interval. So treatment plants, when we use bioaugmentation techniques, we make sure that we continuously monitor it so that the biodegradation is complete by a particular time interval, so that the process is working well and good and it is very, very fast. That is very essential when you are going to add a bioaugmented bacteria. So this is bioaugmentation that we have already seen, and we have also discussed that bioaugmentation can be due to an autochthonous microorganism or an allocthonous microorganisms, or it can be genetically manipulated. So it can be a consortium, it can be a single strain, and it can also be a genetically engineered microorganism or vector. So there are different kinds of bioaugmentation, which is cell bioaugmentation, where you have in a bacterial strain, a consortium, or a genetically engineered microorganism, or a commercial inoculum, which is provided in situ to the contaminated site. So another type of bioaugmentation is gene bioaugmentation. We make sure the vectors carry mobile genetic elements which have specific genes that are capable of producing specific enzymes for the particular pollutant. So that the gene bioaugmentation is going to be very, very specific in nature. Similarly, yet another bioaugmentation is the plant bioaugmentation. So we also know that plants, there are very many plants which are desiccation resistant, which are drought resistant, which are metal resistant, which has a huge wide set of subset of metabolic skills that can be used to remediate a pollutant. So we introduce a specific type of genetically engineered plant or plant with a particular feature to remediate the pollutant. Okay, yet another type of bioremediation is biofilters. So biofilters again, they can be used ex situ, they can be used in situ. So usually biofilters are set into contaminated groundwaters. So they filter, they're treated like pumps. They are transferred into the groundwater table. They're kept inside like bore wells, and these bore wells are set in such a way that they filter the contaminants in the groundwater, and you get treated waters. So here it is a gas stream, but usually in a biofilter, this biofilter is also applicable for filtering toxic gases. So here it is a treated gas stream, but this acts like a borewell, and it is fitted into the soil so that the surrounding contaminated groundwater can be filtered, and it will give you treated water for household or domestic purposes. So the same approach can be done in the treatment plant. So you can use it above soil process. So you can uh, pump the water above ground. You can place an outlet. You can converge water from different resources from underwater tables, and then use biofilters to treat your use biofilters to treat your contaminated groundwater. So it can be used ex situ, it can be used in situ. Uh, it is a very common technique used for treating contaminated groundwater. So the biofilter has an usually applies a bacterial biofilter or a microalgal biofilter. So these days you can see algal treated biofilters very commonly, and they are often used for the treatment of copper and cadmium metal waste. So one of the prolificent microorganisms which is used to treat wastewater and soil for silver nanoparticles is chromatography violation. So this is one remediation technique which has been adapted by different industries in their wastewater treatment plants. Okay, this is again another illustration, a well drawn illustration of how a biofilter is fitted into your soil in situ, and it can be used to treat contaminated groundwaters. So these two slides enlist the different advantages and disadvantages of bioremediation. And so far we have seen, so you can just go through it. These are very easy to go through. And so far we have seen in-situ bioremediation techniques, ex-situ bioremediation techniques which can be applied on both in-situ and ex-situ. So I hope you understood what we have discussed so far. So again, this is a summary of the bioremediation strategies which we have seen so far. So if at all you have any doubts after reading the uh, reading the supplementary material provided along with this presentation, do reach out and ask me your doubts so that we can clarify these in our respective groups. So thank you.